The Active Data Canvas is a visual analytic tool that allows us to explore data within familiar biological contexts. The tool suite is focused on fluid navigation and easy collaboration. The landing page shows current projects and links to the three main tools: the Heatmap View, Pathway View, and the Canvas. Each tool displays project data in a different way. We'll start by exploring our data in the Heatmap View. In this demonstration, we'll be looking at an ovarian cancer dataset. Columns in the heat map represent individual tumors, and the rows represent proteins. Each value in the heat map is the abundance of a given protein in an individual tumor. Dynamic navigation allows you to ask typical questions of the data in real time. For example, what is unique about this cluster of tumors? What differentiates them from the rest of the cohort? By selecting a cluster of tumors, metadata is searched to identify statistically distinguishing features of the cluster. In this case, we see several variables with significant p-values. The List tab shows the identifiers for tumors in this cluster, while the Graph tab allows us to visualize the statistical test. By clicking the new vital status, we discovered that this is a relatively good outcome group, with 50% of the patients still living. Outside this group, patients are deceased at a roughly 3-to-1 ratio. When considering the protein rows of the matrix, people frequently ask, what are these proteins? And what cellular functions are enriched in this group of proteins? This information is easily retrieved from the dynamic interface by clicking on a node for protein clusters. The Canvas automatically searches for functional enrichment using a variety of classification tools like KEGG and Reactome. Browsing various nodes in the heat map, users can quickly navigate the data and ask questions in a self-directed manner. The second tool is a pathway viewer and displays project data on top of KEGG maps. This is typically accessed directly from the functional enrichment test or from the top navigation menu. Red boxes here indicate protein functions for which we have data, in this case MLCK, or the myosin light chain kinase. This pop-up, made with high charts, can be moved and the image can be saved to your desktop. Viewing multiple proteins at once portrays how the entire pathway behaves. The ECM box, or extracellular matrix, shows the expression of many distinct proteins which KEGG classifies to this function. These two tools, the heat map and pathway viewers, allow for fluid and intuitive navigation of biomedical data and are capable of displaying very large datasets in an intuitive and compact way. What makes the Active Data Canvas a truly novel tool is the ability to save and share your thoughts during data analysis through the Canvas view. The Canvas is a pinboard for pieces of data that we want to work on. Data is put onto the canvas with the pin button. Data can be pinned from the pathway view, heat map, or from other applications like Excel or PubMed. Let's open the canvas and see what has happened to our pinned MLCK protein. When you pin data, software assistants proactively search knowledge bases for relevant information. After pinning the MLCK protein to our canvas, two gray boxes automatically pop up with recommendations on potentially useful resources. One box is the NCBI webpage for this protein, the other is a PubMed search using the protein name as a query. Both have links to the external resource. Different types of data trigger different software assistants to gather recommendations. Let's go to the heat map and pin this group we were previously looking at and give it the name of adhesion enriched. Now switching back to the canvas, we see our new node popping up with two recommendation boxes. The first is the quantitative data from the heat map for these proteins, which can be downloaded into Excel or R. The second is the list of enriched pathways, which allows you to navigate back to the pathway viewer. The canvas is designed to be a shareable thought space. All data for a project, including a user's canvas and saved recommendations, is versioned and backed at GitHub. Each experiment or project is a repository where both data and analysis live. Each change is tracked and versioned so that you never lose your place. Using GitHub allows us to seamlessly share the project with specified collaborators or with the world. As each user analyzes data and creates pins, their individual canvas is versioned on GitHub as a part of the project. As a visual analytic tool, the Active Data Canvas lets domain experts explore data within a visual context that is familiar and productive. The tool was created at the Pacific Northwest National Laboratory using laboratory R&D funds.